I'm Povilas, postdoctoral researcher from Lithuania, currently located at Santa Cruz, and my professor is David Dimmer. So, organic compounds, including building blocks of life, may be synthesized in the accretion disk of newly forming solar system and may fall down to Earth as a meteorites and dust. Phosphates, ribose, and all nucleobases were all found in meteorites and under certain conditions may recombine into nucleotides, which are present in all contemporary life. Another essential compound is lipid, which is a membrane-forming amphiphilic molecule. And simple lipids were also found in carbonaceous chondrites, and even though their structure was way more simple, they can form vesicles. So if these compounds fall down to the oceans, they are lost in a dilute buffer. But if they make it down to the surfaces of volcanic landmass, they get a shot at participating in the origin of life. So compounds falling in from space, synthesized in the biogenic atmosphere, cooked up in hydrothermal systems, or through rock weathering, may accumulate on the surface, perhaps finding their way in a volcanic caldera. The key to getting things to interact is concentration. So small, hot spring pools may be a perfect place to cook up some soup of life. And hydrothermal fields of interconnected pools could not only concentrate the compounds, but intermix them between reservoirs, subjecting them to different temperature, pH, elemental and mineral compositions. And for any system to move from simple to more complex one, it must be subject to cycling. And flows of compounds, including building blocks of membranes and polymers, may find their way into a pool, which fills up by a pulse from nearby geyser, dries down, and fills up again. A cycle or an engine is then formed when a polymers between dry lipid layers are synthesized. And during rehydration, they may assemble into membrane-bounded protocells. And the stabilizing effects of polymers and membranes allow the protocells to be tested with some surviving next dry down. As the protocells aggregate together, they form gel-like phase where metabolic cycles can arise. And as a grade fully dries down, the protocells are merging and forming lamellar structures where polymers can resynthesize, template, and allocate. So after many such cycles, simple biological functions can emerge, which, simple biological functions can emerge. These ideas are backed by many studies. And here you can see a model of lasagna of life, where nucleotides are trapped between layers of lipid membranes. And such structures are confirmed by an X-ray diffraction experiment. And the trapping of nucleotides between the layers are beneficial because such structures allow prepolymers to form. And not only that, here you can see that blue lines are with lipid present, and if we compare the depurination reactions, which are degradation of purine nucleotides, in the samples with lipid present, those reactions are really inhibited. If we cook up such lasagna at sufficiently high temperatures, You can see in the last graph at 90 degrees, the nucleotides can really undergo condensation reactions and become, well, oligonucleotides, so RNA or DNA-like molecules. And the most important message from all this presentation is that this is a real system. Like, you can take your prebiotic soup and go to Kamchatka or New Zealand or even in California, the hot spring pool, pour it down, and after a couple of cycles, you will see like real protocells with oligonucleotides, which are of sufficient length to already have enzymatic functions. So my goal for the rest of the year is to use nanopore sequencing to study those sequences to check whether I can find some functional ones. So these ideas and results brought our team to SETI, where we are running a Genesis engine, which is simulation chamber of volcanic landmass with a lot of pools where we can test different conditions. And our goal is to understand what fraction of habitable planets are actually urable. And by urable, I mean in what fraction of habitable planets the life can actually start. Not many people are talking about that, but conditions for life to start will be very different than the conditions which are needed for our life to thrive. Yes? So if you are interested in this project, you can learn more on the SETI website. And I invite you to stop by my poster where we can discuss how we can cook some soup of life and maybe even start a second Genesis. So thank you very much.